So I guess without further ado, I'll start. Today's talk is going to be, is Varroa control possible without chemical miticides? So I think at this point, everyone's more or less aware of what we call the 4 Ps affecting beekeeping, if you will. Parasites, pathogens, pesticides, and poor nutrition. The focus for this talk today will be on Varroa mites in the parasite side here, but I want you all to keep in mind that these things are all very much interconnected, right? Poor nutrition can affect how the bees respond to having pesticides or parasites or pathogens, or being infected with Varroa can increase pathogen loads or make bees less able to deal with pesticides they encounter out in the field when foraging. So these are all kind of interacting factors. But again, for this talk, we're going to be focusing on Varroa mites. So at this point, I think everyone who's worked with bees knows what a Varroa looks like, right? So you can see one here on an adult larva, and this one here on an adult bee, and this one here on a larva. So this is the Varroa life cycle, okay? So an egg is laid, and then at around day 8, Varroa will enter the uncapped brood cell, and it will hide to avoid detection until it gets sealed in. At this point on day 10, the Varroa moves out from hiding, pokes a hole in the larva, and then begins to feed, okay? And by the time we're at here on day 12, this, what we call the foundress mite, this Varroa that first crawled into this uncapped cell, lays its first egg. This is approximately 60 hours after entering this cell. Okay, and the first egg that the Varroa always lays is a male egg. After that, about every 30 hours or so, the foundress mite will continue to lay eggs every 30 hours. And all of these eggs will now be female eggs. Let's see. Here at day 19, after being laid, what we see is that both the first egg that the foundress mite laid, the male, and the females afterwards have finally begun to start to mature, and there is now sibling mating going on, meaning that, you know, brothers and sisters are mating because there's only one foundress mite. There can be mating between mites if, for example, 2 foundress mites enters the same capped cell. But in general, it's a lot of inbreeding that's going on. And then here at point 6, at day 21, this is when the workers emerge with the fully mated Varroa mites, right? Drones take longer to reproduce, right? Or to take longer to develop, right? And so at this point, it takes 3 extra days. And so there are more mature Varroa that are able to leave with drone than they are with workers, right? So all of these that are left here, these are all of the eggs that were laid later on by the foundress mite that haven't had time to fully develop. And these guys will eventually just die off. So what this figure is really showing us is that when Varroa get into these drone cells, their ability to reproduce is much greater, okay? So for the longest time, we thought Varroa fed on the hemolymph of honeybees, but Ramsey's work in 2019 showed us that actually what they are doing is feeding primarily on fat bodies, not the hemolymph of honeybees. And this is really important because fat bodies play the honeybee, okay? Fat bodies can help with the immune functions, pesticide detoxification, storage, synthesis of proteins and waxes, lots of things. So you can imagine if a Varroa is feeding on a honeybee, and then if it's feeding on these fat bodies, it's no longer able to, for example, detoxify pesticides as well, meaning when this worker goes out foraging and encounters pesticides in ag, it is less able to cope with it, right? It also affects immune function. And I think at this point, we're all fairly well aware that Varroa is a vector for many honeybee viruses, right? And so you can imagine if you are a honeybee and you've got a Varroa on you, you are also now more susceptible to all of these other diseases. So Varroa, really bad for many, many reasons. So the question is, how do you know what to do with Varroa, when to do it? So how do you know when to treat? Well, there are lots of methods that people will use to monitor their bees for Varroa levels, but the best and the recommended way of monitoring your Varroa levels is through either using an ethanol wash or a soap wash. There are plenty of articles online that you can find on how to make these ethanol washes or soap washes. I think Randy Oliver on Scientific Beekeeping has a recipe to make a really easy soap wash where it's just, I forget how many tablespoons of like Dawn detergent to water you use. But you can also use ethanol, rubbing alcohol, windshield wiper fluid, whatnot. These are all the, this is a recommended way of getting an accurate count of your Varroa numbers. Some people will use powder sugar shakes, sticky boards, just visual examinations. And while these might get you a rough idea of maybe whether or not you have Varroa, it's not really a good accurate measure of your Varroa populations. And so it's not a good way of knowing when you should treat. In general, there are this guideline of treatment thresholds that can vary throughout the year depending on what phase of growth your honeybee colony is at. So depending on whether your population is increasing, whether it's at its peak, whether it's decreasing or it's dormant, you will have different recommendations of treatment thresholds. But in general, if you are finding anywhere from 2 to 3 mites per 100 bees is when we recommend treatment, okay, to get those numbers below treatment threshold. There are many resources out there for people to help control Varroa and manage. These are from the Honeybee Health Coalition. If you guys have not been to this website, I highly suggest you do. There's all sorts of really great information out there on just honeybees in general, not just Varroa. But if you go to these, they have a really lovely PDF on tools for Varroa management, and they have this lovely Varroa management decision guide, which I will show you real quick. Oh, okay, maybe I'll show you. I have to share. Maybe if I share the desktop, will that work? There we go. You guys can see everything now. So this is the decision management tool. If you go to their website, you can just click to get started. And we won't go through all of this in detail, but I just want to show you all what is available. So it says, welcome, click next. This will help you make decisions as long as you answer these questions honestly. So after sampling, do your Varroa mites levels exceed treatment threshold? So this is our first question, because you should always be sampling before you decide whether or not to treat. It shouldn't just be a given that, oh, it's August, I'm going to treat, right? So you should be sampling and deciding whether or not treatment is necessary. And so, for example, in this case, let's say, yes, my mites are at or above the threshold. You'll click that, and then it asks you, are you open to all kinds of treatments or just one or the other? For the sake of example, I'll say I'm open to both synthetic and naturally derived treatments. Is brood present? So at this time of year, you may or may not have brood depending on where you are. Say, yes, brood is present. Do you have honey supers on? Right now, we don't have honey supers on, so we'll say no. And then you're going to pick at what point in this graph are your bees. So right now in the decreasing population phase this time of year, really heading into the dormant phase. So here are all of the options that you can use that are appropriate for this time of year in your conditions to use for treatment of Varroa. And so if you're open to synthetic chemicals, you can click on these and it'll give you kind of information on what it is, how to treat, What are some restrictions? For example, do not use when colonies are supered for honey. It has to be, it should be better if it's above 50 degrees Fahrenheit, whatnot. And then you can go back and click on these other options if you're curious to know what else is out there. So again, I just think this is a really great tool for people, even if you're not new to beekeeping, to help you realize what options there are out there for treating for Varroa and help you decide what is best for you. So what are some of the other methods of control that we can use to deal with Varroa? Because the question is, can we control Varroa without resorting to these chemicals? So there are some other ways that we can deal with Varroa, including breeding for mite-resistant stocks, making brood breaks, and making splits. However, some of these cultural practices for control can be time-consuming, can be costly, and can really go into your production, right? So if you make a brood break by caging the queen, or if you make splits, you need a new queen. So then you have to find a new queen. That may be expense. Making a brood break will slow down your bees and build up. It may decrease the amount of honey you produce. There are some advantages and disadvantages to each of these. And so as a beekeeper, you kind of have to think about what's right for you in your operation. But these are things that are common ways that we can help keep the Varroa populations down by, again, breaking that brood cycle, because as we learned earlier, brood is necessary for Varroa for reproduction. I'm just gonna show one quick example of a bred line that has been bred for resistance to Varroa. This is the Paul line bees that came out of the USDA, University of Exeter and Louisiana State University. This line has been out for a while, but they recently published, I think a year or two ago, a study on the efficacy of their Paul line compared to commercial lines, because there's a lot of talk of, well, my bees are hygienic, but they don't produce as much or whatnot. And so they looked at these lines and they compared it to commercially available lines throughout the country. And what they found was that their Paul line had lower levels of Varroa than the commercial line, right? In addition, it had lower levels of DWV, DWV A and B, were both lower, as you can see in this blue line, which is their Paul line, compared to this black line, which is their commercial line. In addition, their Paul lines survived longer than their commercial lines. And I don't have this graph here, but they have another graph showing the number of frames and buildup, and they found that their the Paul line was on par with the commercial lines, just as good as building up colony strength and production of honey. So this is kind of showing that, because there was some talk for a while that, yeah, we have Varroa sensitive hygienic bees, but they just don't produce as much, or they're just not as good pollinates. But in this study, they show that their line is just as good as commercially available lines for production and for pollination and all of that. So there's a lot of hope here in breeding for Varroa resistant lines. So you can do breeding traditionally, right, through setting up mating yards, or you can do a much more targeted approach, which is what we kind of want to do when we're looking for breeding for bees to make it go faster, right? Because we want, we needed Varroa resistant bees yesterday. And so there are different ways of selecting for bees, and Sue Kobe, if you aren't aware, is one of the international well-known people in honeybee insemination. We had a workshop this summer on cryopreservation and insemination. But this is one of the techniques that a lot of bee researchers here at WSU and around the world are starting to use to create specific lines of bees, right? So you can really control the genetics if you do instrumental insemination. There are other ways of selecting bees that you want to mate, including this study came out of Foster's lab in Canada, I believe, and they've done selective marker-assisted selection to help with breeding. And so they developed protein biomarkers to help them find traits that are associated with hygienic behavior so that they can select the correct bees that they want to breed. So again, help making this breeding process more efficient and better. So that's really a lot of the work that's been done on the making mite-resistant stocks. There are plenty of other labs and queen breeding companies out there that are looking into and creating their own lines of mite-resistant stocks. So they are out there. Another method of control for Varroa mites that doesn't require any sort of chemical input is drone brood trapping, right? So Varroa mites preferentially like drone broods because they take longer to develop, so they have the ability to produce more offspring. However, drone brood trapping is a very time-consuming process, right? You're inserting frames into your hives and it's not only time consuming, but it's a resource consuming process, right? You're think about all of the resources the colony has to put into raising and developing these poor drones that you are going to throw out in the end anyway. And this is a big thing, is because this is time sensitive and time consuming, if you do not pull your drone brood frame before the drones emerge, what you've done is just bred a bunch of Varroa. Right? And we want to be beekeepers, not Varroa keepers, right? And so mechanical, this form of control, this drone brood trapping can be effective, but it requires you to be on a very strict schedule and to, you can't be a day late and let these drones emerge with all these Varroa. So the question is, again, is Varroa control possible without chemical miticides? And my answer to that is maybe. We're not there yet, but I like to think of myself as an optimist, and I think that it's possible and it's in the future that we can be able, I think the long-term solution is to be able to breed bees that are able to deal with Varroa. So as of right now, it's not possible, but in the future, I believe it is. But for now, what we must do is we must monitor our hives regularly and treat when necessary, right? Not prophylactically, not on a calendar, but treat when you exceed threshold. So as far as these methods of chemical controls that go out there, I showed you that website earlier on the Honeybee Health Coalition. They have a lot more data and descriptions on each of these different chemicals, but in general, they're kind of grouped into hard or soft chemicals, or synthetic and organically derived chemicals, if you will. These are these two general class of chemicals that we have for use against Varroa. So again, I want to emphasize that while some of these cultural mechanical practices may work for a bit, if they don't work, and you know this because you're monitoring regularly, you have to move on to treatment. Right? You can't just let it go, because then your Varroa populations will just get out of control. And there's lots of evidence saying that if you do nothing when you exceed threshold, it's just kind of like a death sentence for your colony. So with all of these different kinds of chemical controls out there, there are some drawbacks, not just because they're pesticides in the environment, but also lots of evidence of Varroa being resistant to these chemicals, meaning they aren't as efficacious anymore and they aren't working as well as they used to. And this has been going on for a long time. This is from Jeff Pettis at the USDA from 2004, describing how Varroa is already having some resistance to coumaphos. This was just, oh my gosh, 2020 was not last year. I guess that was 2 years ago. But, Amitraz resistance within commercial operations. This came out of the USDA lab in Baton Rouge, right? And this isn't something that's happening just here in the US. It's not isolated to us here. This is a worldwide problem, right? So this is going on in this article in Israel, here in the UK, in Mexico, South America. It's everywhere, right? So resistance is not unique to us. It's not a problem that's an American problem, if you will. It's a honeybee problem. These Varroa mites that are becoming resistant to the most common chemical treatments that we have available. And so a lot of the research that been done here at WSU with myself, Dr. Nager, and Dr. Shepard is looking at the use of fungi to act as a biocontrol agent to control Varroa mites. And I throw this picture out there because everyone always thinks of fungi as these delicious edible fungi that we're all familiar with. And just on a side note, chanterelles and hericium are both delicious and they're both out right now. So if anyone's interested in fungal foraging, now is the time to go look. Anyway, fungi come in all shapes and sizes, right? They're not just these mushroom bodies. There are many that are just like these mold-like things. And many of these are what we call entomopathogenic fungi, meaning they infect and kill insects. So the fungi that we have been working on with is called a Miticides. It's an entomopathogenic fungi, like I said, and it's native, which is really awesome because I said that this is a worldwide phenomenon, right? It's not just something that's isolated to us here in the US. And so the hope is to use a fungal biocontrol agent that is also has a worldwide distribution, right? I think all of us have heard the classical biocontrol stories that have gone out of control, right? You introduce one thing to control one thing and then it gets out of control. So you have to, introduce a snake to eat the frog, to eat the whatnot. But in this case, this fungus is actually found throughout the world, right? It's found on all continents, I think, except Antarctica. So we're not introducing anything new. Here in the US, it's been approved EPA safe for human contact and consumption. And early USDA studies showed that it was not able to effectively infect or kill honeybees. So there's safety with honeybees as well as has been shown through the USDA. And these were some initial studies using Miticides in field trials, and they found that there was initial efficacy for Miticides, but that there were some issues with the fungus. Right? It had the ability to kill Varroa. However, it had issues with thermal tolerance. So honeybees thermoregulate their hives to stay warm, right? At a much warmer than the ground, right? And this fungus is normally a soil-dwelling fungus. So you can imagine that their temperature that they're used to growing and living in is much cooler than, say, 90-some degrees, which you will typically find in the center brood chamber of a hive. So when you try to grow this fungus at cool temperatures versus temperatures that it will encounter in a beehive, you can see that the growth of this fungus is not very good, right? So this is at cool temperatures, this is at hive temperatures, and you can see that it's very much stunted and not looking very, if you will, healthy. So what we did was we did a whole selection process, what we call directed evolution, to improve its biocontrol efficacy. So this idea of directed evolution or artificial selection is a very old idea, actually. You guys may be familiar with the selection from brassica, right? Our old cabbage relatives. Depending on what you select for, if you select for terminal buds, in this case, you end up with cabbage. If you select for leaves, you end up with kale. If you select for lateral buds, you end up with Brussels sprouts, right? These are all different variations derived from the same common ancestor based on what they had, the traits that they selected for. So we're taking the same principle of selecting for specific characteristics to end up with a strain that has what we want. So in our case, what we did was we started off with one strain of spores. We induced, I guess, mutations, if you will, by stressing it out. And what we did was we stressed it out using temperatures. We stressed it out using a nutritional stress and a oxidative hydrogen peroxide stress. And there's a lot of efficacy saying if you're stressed out nutritionally, you are cross-protected against other stresses, including like heat or UV exposure, whatnot. So in the lab, we stressed out these spores, induced some different mutations. We selected the best performers. We did it again. We did this over recursive rounds over and over again, always selecting the best strains. Simultaneously to our laboratory selections, we were also doing these selections out in the field by putting our strains in actual beehives, infecting live Varroa, figuring out which strains were the most effective out in the field, selecting those best strains, and then again, putting it through this selection process. So there were 2 different selections going on, right? In the lab and in the field. In the lab, we were selecting for thermal tolerance, and in the field, we were selecting for virulence. And so there's 2 forms of selections were going on simultaneously. So this is our field work. This is the lab work. And in the end, there were a lot of treatments and a lot of screening done. I think at one point when we were halfway through, it was over 30,000 mites that were screened and selected. And in the end, what we see is our parental strain versus our final selected strain has very different phenotypic characteristics. You can see here the starting strain. You can see that it's very weak growth, and you can see what these, I guess, little candy like Mike and Ike little cone things are, hot dog shaped things, are spores that haven't germinated. Okay. And in our parental strain, when you grow them at hive temperatures, what you see is a lot of spores don't germinate. You have a low percentage. And the ones that do, they're what we call germination tube here is very short and stunted, which is kind of analogous to the health of the spore, if you will. After the selection process, what you see is a higher proportion of spores germinating. In addition, the ones that do germinate, their germination tube is longer, showing that it is behaving better and is a bit more healthier at these high temperatures. And when we look at the final data, what we can see is that the treatment duration is increasing, meaning that this treatment is more effective for longer periods of time, and the hives that were treated with this metarhizium are living longer. We also did a study where we looked at the efficacy of this fungus compared to oxalic acid. And what we found was it was just as effective as keeping the mite levels below treatment threshold as oxalic acid. And oxalic acid is higher here, but it is still below treatment threshold. So prior to this, what we were doing was we were growing a lot of our metarhizium, this fungus, on agar dishes, which is great for in the lab, but it really isn't a very practical solution for growing it up in bulk and for treating lots of hives. And so what we did was we looked at what was the best bulk substrate to use for growing. We did a survey of several different types of grains and different water scenarios and hydration, moisture. And we found that growing fungus in these grow bags with rice was the most economically viable and also had the best production. We also looked at how best to deliver now, because prior to this, like I said, we were growing these on Petri dishes. But now that we're growing it on grain, how do we get the grain to the bees? And we did a lot of experimenting, trying to figure out what the best method was. We looked at these fiber bags, but these ended up being a little too tall and required spacers to be put between the top box and the lid or supers or whatnot. What ended up doing was that started to disrupt the behavior of the bees. They didn't want to go up into the top box if there was a super up here because of this space. So we had to come up with a different solution. In the end, we decided to remove these spacers and instead come up with these bigger, flatter bags of grains to be used to treat the hives. We also looked at creating these grease patties, if you will, of sugar, Crisco, and the colonized grains and using them to treat the hives. And we found that both the grease and the bags seemed to work okay, as just as effective as oxalic acid in the controlling the Varroa population numbers. And what we're doing right now is we're working with the Office of Commercialization to get this strain out in the public. We are currently in the process of talking with a couple of different companies. I don't want to, it's all very preliminary, so I don't want to say too much, but we are talking with a couple of companies who are interested in collaborating and producing this strain for commercial sale. So we're kind of at that point right now. I just want to mention a few other things that are going on. So I think I've mentioned this before, but Metarhizium naturally produces oxalic acid. And as many beekeepers know, oxalic acid is a treatment that we use to control Varroa mites. So there was a question of, is there a synergistic effect between Metarhizium and oxalic acid? And this was done by master student Fatima, where she found that, yes, in fact, there is a synergistic effect. If you put these spores in oxalic acid, you can end up with better control with less oxalic acid. So the idea is that maybe we can reduce the amount of other chemicals we use if we combine this with Metarhizium, right? Maybe we don't eliminate it entirely, but maybe we can reduce the input that we're putting into the hives. This was a study done by Riley. She's currently in Purdue now working with Brock Harper in their entomology department as a master's student. But she looked at the health effects of Metarhizium on honeybees and longevity. And what she found was that bees treated with Metarhizium, at least in cages, lived and survived longer than bees that didn't have the fungal spores. That was the only difference. They didn't get any difference in nutrition or anything else. Just having that treatment, for whatever reason, seemed to increase their longevity. So she did some qPCR work to look at, what was going on, and what she found was that in some cases it seemed like there were some immune function genes that were being upregulated in the bees that had fungal treatment, for example, dicer. And in this case, dicer is an integral part in the RNA degradation pathway, especially viral RNA. So there could be some interesting effects with the Metarhizium, not just on dealing with Varroa, but also perhaps helping boost the immune system and help dealing with viral infections. Anna Webb was Dr. Brandon Hopkins' student. She graduated last year, but she looked at combining Metarhizium with indoor storage treatments to see if a break in the brood cycle could help make these treatments more effective. And then this final project I'm going to mention, and I think Dr. Hopkins will be talking more about this. I think he'll have an entire talk on this. Was that M. Rendelman? She graduated. She did graduate just this last May. And one of her projects was looking at a formulation of oxalic acid that was formulated onto strips to be more stabilized. And they found that the stabilized oxalic acid was really good at keeping the number of Varroa mites down. So while this may not be a non-chemical control, it's showing us that the chemicals that we do have out there, there are ways maybe that we can make them better and more efficacious. And so I guess I'll end with the question again, is Varroa control possible without chemical miticides? And as much as I wish I could say yes with my, wholeheartedly, right now we're at that maybe. There are lots of things that we can do, I think, to minimize the amount of chemical miticides that we use, but we aren't able to go completely chem-free right now. But I am hopeful that with breeding and with these new research that we're doing, that it's a possibility. And again, I can't emphasize this enough, but it's really important to monitor your hives regularly and treat when necessary, not just prophylactically, because I think a lot of the resistance issues we have, can come down to lots of different misuse of some control measures. And I guess I'll end this talk with a video. This is Varroa that we've dipped in our fungal spore solution.
Jennifer, hold for just a minute. The last few slides, we're having some technical issues here. So go ahead and hold on the video if you don't mind. And maybe we can take questions while Brandon tries to do the AV. So we got to see a lot of your slides, but these ones near the end, we just listened to your mellifluous voice. So,
I hope you enjoyed the voice.
yeah, no, it was great. It was really great. Can we get, are there any questions? And I'll just repeat them for Jennifer.
Okay.
It would be before it's commercially available.
Jennifer, the question was, how long do you think it would be before it could be commercially available?
That's a great question. And Steve, I don't know if you have thoughts on this too, but I know that we are, myself, Dr. Nager and Steve, we are working with and talking with some other companies right now, trying to figure out, I guess, a timeline and getting things started as far as making things commercially available. But things are kind of still in the talking stages.
There is a preliminary patent file. Yes.
For application in the hive, I'm wondering if it's possible to grow it on rice paper sheets.
Okay, Jennifer, the question was for application in the hive, is it possible to grow it on rice paper sheets?
It's a great question. I haven't attempted that. Maybe, but I imagine that the rice paper sheets might be, they would dissolve a little too quickly, right? And I don't know if it would stay together in that same way, but that's definitely something that we could explore.
It might not have enough nutrition in
Yeah.
paper. Awesome.
Right? I'm not really sure what rice paper sheets are made out of, other than rice.
Questions? Are there any other questions? Yes.
Hey, has anybody looked at, basically viral pathogens instead of just fungal pathogens for Varroa?
So Jennifer, the question was, has anyone looked at viral pathogens rather than just fungal pathogens for controlling Varroa?
Oh, not that I know of.